Hi, I'm Annie.
Hi, I'm Edward.
Hi, my name is Sandia.
Hi, I'm Victoria. Sandia is up late studying for AP Bio. This is her 6th Celsius of the night. Uh-oh, what's happened here? Sandia has been mutated. Let's take a closer look. Her cells are dividing uncontrollably. Let's see a normal cell for comparison. Normally, the cell passes through the G1, G2, and N checkpoints so the cell doesn't divide uncontrollably. After growth, protein synthesis, and other regular cellular processes in the G1 phase, it goes through the checkpoint. First, the G1 checkpoint checks if there's enough nutrients, that DNA is all correct, and if there are any growth signals going through. Then, DNA is replicated in the S phase. The G2 checkpoint checks to see if the DNA was replicated correctly and if the cell can undergo mitosis. If, and only if the cell passes these checkpoints, the cell undergoes mitosis. During prophase, chromosomes condense and the nuclear membrane breaks. The centrosomes move to opposite ends and form the mitotic spindles. In metaphase, the chromosomes line up against the metaphase plate and spindle fibers attach from each side of the cell. Furthermore, the chromosomes are now condensed into their X shape. In anaphase, the microtubules from the spindle pull each chromosome into two sister chromatids toward opposite poles. In telophase, two daughter nuclei form around each cell's chromatids. The cleavage furrow then forms and splits the cell into two.
I can't tell why your cells are dividing uncontrollably. Guess I'll go take a closer look. Okay, what the heck?
Does this mean I'll die?
What? No, of course not. How do I fix this?
If I may.
Yes, Miss Narrator?
Caffeine is inhibiting the kinase, an enzyme that activates the p53 gene. Now damaged DNA is being replicated and passed on to the daughter cells. In normal DNA replication, a strand of DNA is unwound by helicase, separating the strands at the replication fork. The DNA separates into a leading strand and a lagging strand. Primase puts RNA primers on the strands to signal the start of replication. During DNA synthesis, the 5' end gets synthesized first, and replication ends at the 3' end. New bases are paired according to the base pairing rule, adenine with thymine and guanine with cytosine. The leading strand is synthesized heading toward the replication fork in one continuous strand. The lagging strand is synthesized in separate pieces, called Okazaki strands, away from the replication fork. Afterward, DNA polymerase removes the leftover primers and fills in the gaps with more DNA. Ligase then seals all of the new fragments together into one continuous, unbroken strand. In the end, each new strand has one strand of old DNA and one strand of new DNA, making replication a semi-conservative process. Sandia's DNA can't stop replicating, and it's adding new mutations every second. The p53 gene suppresses tumors and triggers apoptosis in the event of uncontrolled division like this. It's involved in every checkpoint of the cycle to prevent this exact situation.
Kinase is an enzyme necessary for the proper activation of the p53 gene, but Sandia has had so much caffeine that the particles are denaturing her enzymes. Her cells aren't getting the signal to stop dividing.
Okay, I got it.
Got what?
Your tumor.
Tumor?
No worries, it was a benign tumor caused by uncontrolled cellular growth spurred on by your hopeless caffeine addiction. You should probably drink less.
What? I can't do that. No!